Some of the most advanced and exclusive systems in existence are not found in luxury machines or elite technology. They are operating inside your body right now. Let's start silently measuring, testing, and deciding what stays and what gets removed. Every second, a hidden system is evaluating your cells with a level of precision that rivals engineered systems, and most people have no idea it's happening. In this video, you're going to understand how your cells maintain order against constant breakdown. That's how fasting changes this process at a deep biological level, and why a specific time threshold can completely shift your body from growth into repair. You'll see how energy, physics, and biology combine into one system that continuously rebuilds you from within. Before we go deeper, make sure to subscribe to the channel and turn on the bell icon so you don't miss more informative and educational content like this. Right now, inside your body, every mitochondrion is being monitored. Each one is running a continuous electrical test. Your cells are checking whether these tiny structures are maintaining a specific voltage, about 180 millivolts across their inner membrane. That electrical charge is not just a detail. It's the core signal that tells the cell whether a mitochondrion is functioning properly or starting to fail. If a mitochondrion drops below that threshold, it doesn't get ignored. It gets marked, it gets isolated, and then it gets removed. The cell identifies it, surrounds it, breaks it down, and recycles its components into something new. This is not something that starts when you decide to fast. This process is always running in the background. It never stops. What fasting does is not create the system. It accelerates it. And it does so in a way that can be measured at the molecular level. When the body is constantly fed, this system is suppressed. When food intake stops, the system becomes more active, more aggressive, more efficient. This mechanism was important enough that it earned a Nobel Prize in 2016. But even after that discovery, most explanations still don't fully capture what's really happening, especially when you look at it through the lens of physics. Because this is not just biology, it's thermodynamics. To understand why your cells need to break down parts of themselves, you have to start with a simple truth. Our living cell is an extremely ordered system in a universe that constantly moves toward disorder. That's the second law of thermodynamics. Everything naturally tends toward chaos unless energy is used to maintain structure. Your cells are fighting that process constantly. Inside each cell, there are precise ion concentrations, carefully folded proteins, and highly organized structures that all need to stay exactly as they are. But at body temperature, nothing stays stable on its own. Proteins are constantly vibrating due to thermal energy. A single protein made of hundreds of amino acids can shift into incorrect shapes thousands of times per second. Most of these errors correct themselves instantly, but some don't. Some become stuck in the wrong form. And when that happens, the cell has to act. Damaged proteins and mitochondria accumulate damage over time, interfering with normal function. Every structure in the cell is under constant pressure from entropy. When food is abundant, the cell prioritizes growth. But when intake stops, the situation changes completely. Around 12 to 16 hours into a fast, the system hits a threshold. It doesn't slowly adjust. It switches operating modes. Like water freezing, the cell moves from growth to repair. It stops building and starts auditing, identifying what is no longer working, recycling it, and rebuilding with better components. This is controlled by a biological switch, mTOR and AMPK. When energy is low, AMPK inhibits mTOR, turning off the growth signals to activate deep cellular restoration. This is not starvation, this is optimization. This is where the switch happens. It's not gradual, it's not partial. Below a certain threshold, mTOR remains active and autophagy stays suppressed. But once AMPK crosses its activation point, it shuts down mTOR's signal, releasing the brake on autophagy. The system flips, repair begins. This is why time matters in fasting, but not in the way most people think. The benefit doesn't increase smoothly with each hour. Instead, it depends on whether you cross the threshold. Someone who stops eating at 8 p.m. and eats again at 10 a.m. completes about 14 hours of fasting. That may not be enough to fully activate autophagy, but waiting until noon Just 2 more hours can push the system past the threshold. And those 2 hours are not equal to the previous 14. They are the difference between one state and another. This is what defines a phase transition. Just like water doesn't slowly become ice, but changes state at a precise point. The cell doesn't slowly increase repair, it switches into it. And this exact mechanism was not originally discovered in humans. It was discovered in yeast. A scientist named Yoshinori Osumi was studying yeast cells under nutrient deprivation, specifically nitrogen starvation. What he observed was something unusual, structures forming inside the cell that had not been clearly understood before. These structures were membrane-bound compartments filling with cellular material. To understand them, he engineered yeast cells that could not break down the contents of these compartments. As a result, the structures accumulated and became visible. What he was seeing were autophagosomes. These are the structures that engulf damaged components inside the cell and deliver them for recycling. Over time, Osumi identified the genes responsible for this process, now known as ATG genes, autophagy-related genes. These genes control how the autophagosome forms, how it selects targets, and how it delivers them for degradation. At the time, this was considered a minor housekeeping function, but it wasn't. It was a fundamental survival system. And what made this discovery even more important is that the mechanism is conserved across evolution. The same system found in yeast exists in human cells. The same genes, the same machinery, the same logic. A process that evolved over 1 billion years ago is still operating inside your body right now. This is why the Nobel Prize in 2016 recognized this discovery. Not because it introduced something new, but because it revealed something that had always been there, operating silently and continuously. And once the switch activates, the next stage begins. The cell starts building a structure designed specifically for recycling. This structure is called the autophagosome. It is not a simple container. It is a highly engineered system built from a lipid membrane that must bend, expand, and seal around damaged components. And this process is not easy from a physics perspective. A membrane naturally prefers to remain flat. Bending it into a curved structure requires energy. The more curvature you create, the more energy it costs. This is described by physical principles governing membrane behavior, yet the cell does this repeatedly. Autophagy is the cell's rapid recycling system, turning flat membranes into closed spheres to engulf damaged organelles in just minutes. Controlled by specialized proteins, these structures deliver their contents to lysosomes, where they are broken down into reusable amino acids and lipids. This is not random destruction. When targeting mitochondria, the process is called mitophagy, a selective system that monitors organelle health. The cell measures one key parameter, voltage. A drop in electrical potential signals that a mitochondrion is failing and marks it for removal. Proteins like PINK1 and Parkin detect this failure and tag the damaged structure, making it visible to the cell's cleanup machinery. Once tagged, the autophagosome captures the mitochondrion with remarkable precision, ensuring the cell remains clean and efficient. It is a precise, intelligent system that clears out dysfunction and replaces it with usable material. This is the science of cellular survival. It is targeted removal based on a single measurable parameter, voltage. The elegance of this system is in its simplicity. The cell does not need to evaluate hundreds of variables. It uses one signal as a proxy for overall function. If the mitochondrion cannot maintain its electrical charge, it cannot perform its role. And so, it is replaced. This process is running continuously. Every mitochondrion in your body is under constant surveillance. The ones that maintain their charge remain. The ones that fail are removed. But here's the critical detail. In a constantly fed state, this system is limited. Even when damaged mitochondria are identified and tagged, the machinery responsible for removing them, autophagy, is partially suppressed by mTOR. The signal to recycle exists, but the execution is slowed, so damaged mitochondria accumulate. They produce less energy and more reactive oxygen species, which increases cellular stress and damage over time. Fasting changes this. When the mTOR brake is released, the autophagy system becomes fully active. The backlog of damaged mitochondria is cleared more efficiently. The cell removes weak components and replaces them through mitochondrial biogenesis. This renewal process is partly driven by a regulator called PGC1 alpha, which is activated during fasting and promotes the creation of new mitochondria. The result is a shift in the entire population. After a period of fasting, the mitochondria inside the cell are, on average, healthier, more efficient, and less damaged. This is not a minor adjustment. It is a full quality control cycle, and it changes how you understand what the cell is doing, because the decision is not chemical, it is electrical. The cell evaluates its energy systems based on voltage, using the same physical principles that govern nerve signals and muscle contraction. This is physics applied at the cellular level. And there's another layer to this that makes the process even more interesting. Mitochondria are not originally part of your cells. They were once independent organisms. Around 1.5 billion years ago, ancient cells absorbed bacteria capable of producing energy. Instead of destroying them, they formed a symbiotic relationship. Over time, these bacteria became mitochondria. But they never lost their identity completely. They still have their own DNA. They still use machinery similar to bacteria. And they still operate using electrochemical processes that resemble their original function. Every cell in your body contains hundreds or even thousands of these structures. They produce most of the energy you use. And when your cell evaluates them, it is essentially deciding which of these ancient systems are still functioning and which need to be replaced. This is not just maintenance. It is management of a symbiotic relationship that has existed for over 1 billion years. When a mitochondrion fails, the cell removes it and replaces it with a new one. The system ensures that only the most efficient structures remain active. Fasting accelerates this process. It increases the rate at which damaged mitochondria are cleared and new ones are generated. So what you end up with is not the same system you started with. It is improved, cleaner, more efficient, and better adapted to produce energy. This is where the process moves beyond simple repair. It becomes optimization. And while this system handles large structures like mitochondria, there is another system working at the same time, handling a completely different type of problem. Not large components, but individual damaged proteins. And that system operates with a completely different mechanism. While autophagy handles large structures, entire organelles, clusters of damaged material, there is another process running continuously in the background, working at a much finer level. Instead of engulfing bulk material, it focuses on individual molecules. This system is called chaperone-mediated autophagy, or CMA. It does not rely on building membranes or forming large vesicles. It works one protein at a time. Inside the cell, proteins are constantly being monitored. Some of them carry a specific sequence, a kind of built-in tag that signals they can be inspected and, if necessary, removed. This sequence allows a chaperone protein known as HSC70 to recognize them. When a protein becomes damaged or misfolded, HSC70 binds to it and escorts it directly to the lysosome. But unlike macroautophagy, there is no engulfing structure. Instead, the protein is unfolded and threaded through the lysosomal membrane, one molecule at a time. Inside, it is broken down into its basic components and reused by the cell. This is an entirely different level of precision. It's not bulk recycling. It's targeted removal at the molecular scale. Under normal conditions, this system is responsible for a significant portion of protein degradation inside the cell. It ensures that damaged proteins are handled immediately before they can accumulate, and that matters. Because if misfolded proteins are not removed quickly, they begin to stick together. These aggregates grow, become resistant to breakdown, and interfere with cellular function. CMA prevents that from happening. It acts as a continuous cleanup system, handling damage as soon as it appears. But here's the problem. As the body ages, this system becomes less efficient. The number of receptors on the lysosomal membrane decreases, reducing the rate at which damaged proteins can be processed. The throughput drops, and when that happens, misfolded proteins begin to accumulate. At that point, the cell has to rely more on bulk systems like macroautophagy to remove them. But that approach is less efficient for handling individual molecules. It's like the difference between picking up small pieces of waste as they appear versus waiting for them to pile up and then removing them all at once. The second method works, but it's slower and less precise. Over time, this accumulation contributes to the formation of protein aggregates associated with aging and neurodegenerative conditions. So now you have two systems working in parallel. One handles large structures. The other handles individual proteins. They operate differently, respond to different signals, and decline at different rates. And this distinction is important because fasting primarily enhances the bulk system, macro-autophagy. It does not fully restore the precision system. That means while fasting helps clear larger damage and improves overall cellular condition, it does not completely solve the issue of declining molecular level cleanup. Both systems matter. Both are necessary, and both are influenced by different factors. Now shift your focus to the brain, because this is where another critical change happens during fasting. After a certain period without food, the body begins to run out of stored glucose. At that point, the liver starts producing an alternative fuel source, ketone bodies. One of these molecules, beta-hydroxybutyrate, becomes especially important. It crosses into the brain and begins to supply energy directly to neurons. That alone is significant. But what makes this molecule unique is that it does more than just provide fuel. It also acts as a signal. Beta-hydroxybutyrate influences gene expression inside brain cells. It interacts with enzymes that regulate how DNA is read, effectively changing which genes are active. Specifically, it keeps chromatin, the structure that holds DNA, in a more open state, allowing certain genes to be expressed more easily. Among these genes is BDNF, or brain-derived neurotrophic factor. BDNF plays a key role in learning, memory, and the formation of new neural connections. It supports plasticity, the brain's ability to adapt and reorganize. At the same time, other genes related to stress resistance and cellular protection are also activated. So this single molecule is doing two things at once. It is fueling the brain, and it is reprogramming it. This is not a common combination. Most substances either provide energy or act as signals, not both simultaneously. But here both functions are happening through the same molecule. And this has important implications, especially for aging. As the brain ages, levels of BDNF decline, neural plasticity decreases, the ability to form new connections slows down. But during fasting, the increase in beta-hydroxybutyrate activates the very pathways that counteract these changes. This helps explain why many people report improved mental clarity during extended fasting. It's not just subjective. There is a molecular basis behind it. The brain is operating on a more efficient fuel source while simultaneously activating pathways that support adaptation and resilience. This is not deprivation, it is activation. The system that turns on when food is removed is not designed to weaken the body. It is designed to optimize it under different conditions. And that realization changes how you interpret the entire process, because what appears from the outside as a lack of input is, internally, a shift in strategy. The body is not shutting down, it is reorganizing. And now, with all of these systems working together, bulk recycling, targeted protein removal, mitochondrial quality control, and metabolic signaling, the full picture begins to form. But one final question remains. What part of this is fully proven and what is still being explored? And that question matters more than it seems. Because when you look at a system this complex, spanning physics, biology, and metabolism, it's easy to assume everything is fully understood and precisely defined, but that's not the case. Some parts of this process are firmly established. Others are still being explored. The mechanism of autophagy itself is not in question. It has been observed, mapped, and studied in detail. The genes involved, the structures formed, the signals that activate it, these are well documented. The work that earned the Nobel Prize confirmed that this system is real, conserved across species, and essential for cellular maintenance. That part is settled. But when you move from mechanism to application, especially in humans, the picture becomes less precise. We know that fasting can activate autophagy. We know that nutrient deprivation reduces mTOR activity and increases AMPK signaling. We know that these changes shift the cell from growth into repair. But what we don't yet know with certainty is how specific fasting protocols translate into consistent, measurable outcomes across different individuals. The commonly discussed 16-hour fasting window is based on the idea of crossing that threshold. But the exact point at which that threshold occurs can vary. It depends on metabolic health, it depends on age, it depends on activity level, diet, and even genetics. For one person, 16 hours might be enough to trigger significant autophagy. For another, it might not be. That variability is important because it means that while the mechanism is real, the optimal way to apply it is still being refined. Some of the strongest human data comes from structured approaches like fasting mimicking diets, which extend beyond simple daily fasting windows and show measurable improvements in markers related to aging, inflammation, and metabolic health. But those protocols are more demanding, and they don't map directly onto the simpler patterns that are often promoted. So there are two truths that exist at the same time. The mechanism is valid. The optimal human application is still being defined. Ignoring either one leads to misunderstanding. Saying that fasting will definitely produce a specific result in every case is an overextension. But saying that the underlying biology is uncertain is equally inaccurate. The system exists, the switch exists, the response exists. What varies is how consistently and how strongly it is activated in real-world conditions. And then there is the larger context, the principle that sits behind all of this, the second law of thermodynamics. This law does not depend on biology. It applies to every physical system. Everything tends toward disorder unless energy is used to maintain structure. Your cells are constantly resisting that process. Every membrane, every protein, every organelle is under continuous pressure to degrade. And maintaining order requires energy. When energy is available, the cell can build and repair at the same time. But when energy becomes limited, it must prioritize. It cannot do everything equally, so it makes a decision. It shifts from expansion to maintenance, from building outward to repairing inward. Fasting is one of the conditions that triggers this shift. It does not create a new process. It changes the balance of an existing one. It removes the constant input that keeps growth signals active and allows repair systems to operate at a higher level. And when that happens, several things occur at once. Autophagy increases, damaged mitochondria are cleared, new mitochondria are generated, protein aggregates are reduced, and the internal environment becomes more efficient. This is not a single pathway. It is a coordinated response, a system-wide adjustment to changing conditions. But modern patterns of eating can interfere with this. If food intake is spread across most of the day, starting early in the morning and continuing late into the evening, the fasting window becomes too short. The threshold is never crossed, the switch does not activate fully, and the repair processes remain limited. This does not mean that food itself is harmful. It means that constant feeding maintains a state where growth signals remain active and repair signals stay suppressed. The system is designed to alternate between these modes. But if one mode dominates continuously, the balance is lost. Damage continues to accumulate, even if slowly, and the mechanisms that would clear it are not fully engaged. This is not a recommendation, it is a description of what the system requires to operate as it was designed. The threshold exists, the switch is binary, and the conditions on either side of it produce different outcomes. Understanding that distinction is critical, because it shifts the conversation from simple advice to underlying principles. And once you understand the principles, you can begin to see how the system behaves under different conditions. But there is still one final layer to consider, not just how the system works, but what it ultimately means for the long term. And that final layer brings everything together. Because once you understand how these systems operate, how cells monitor damage, how they switch between growth and repair, how they recycle and rebuild, you begin to see that this is not just a collection of isolated processes. It is a unified strategy, a strategy built around one fundamental challenge, maintaining order in a system that naturally moves toward disorder. Everything you've seen, autophagy, mitophagy, protein surveillance, metabolic shifts, is part of that single objective: to preserve function, to maintain structure, to keep the system operating despite constant pressure to degrade. And the key point is this: these processes are always running. Your cells are not waiting for fasting to begin repairing themselves. They are doing it right now, continuously, at a baseline level. Proteins are being monitored. Damaged components are being removed. Mitochondria are being evaluated. But the intensity of that work changes. When nutrients are abundant, the system prioritizes growth. It builds, expands, and maintains at the same time, but the focus remains on construction. When nutrients are limited, the priority shifts. The system turns inward. It reduces external building and increases internal repair. Fasting is one of the conditions that triggers that shift. Not because it introduces something new, but because it changes the balance between two existing modes, growth and repair. And that balance matters over time, because small differences in how damage is handled can accumulate. If repair processes are consistently limited, even slightly, damage builds gradually. Misfolded proteins persist longer. Mitochondria operate less efficiently. Cellular systems become more stressed, not immediately, but progressively. On the other hand, when repair is periodically prioritized, that accumulation is reduced. Damaged components are cleared more effectively. The system resets to a more efficient state. Again, not instantly, but over time. This is where the long-term implication becomes clear. The body is not static. It is constantly updating itself. Cells are replaced, components are recycled, structures are rebuilt. What you are at one moment is not exactly what you are later. The system is dynamic, and the way it is managed determines how well it continues to function. Fasting interacts with this dynamic system by creating conditions where repair becomes dominant, even if temporarily. It allows processes that are normally secondary to take priority. And that shift, repeated over time, can influence how the system maintains itself. But it's important to stay precise. This is not a guarantee of specific outcomes. It is not a single solution. It is a mechanism that responds to conditions. And like any mechanism, its effects depend on how and when it is activated. What is clear is that the biology is real. The cellular switch exists. The repair systems exist. The energy signals that control them are well understood. What remains uncertain is how best to apply this knowledge in a consistent, individualized way. And that distinction is important because it separates what is proven from what is still being explored. You can be confident in the existence of the system. You should be cautious in making precise claims about its outcomes. That balance reflects the current state of understanding, and it brings us back to the central idea. Your body is not passively aging. It is actively managing itself. Every second it is measuring, repairing, replacing, and adapting. The second law of thermodynamics continues to push toward disorder. Your cells continue to push back. Fasting does not change that battle. It changes how the body responds to it. It shifts resources toward maintenance. It accelerates cleanup. It allows the system to focus on restoring function rather than expanding it. And that shift, simple in concept, complex in execution, is what defines the process. Not a shortcut, not a guarantee, but a condition under which the body operates differently. More focused on repair, more engaged in renewal, more aligned with the systems that have been maintaining life at the cellular level for billions of years. And once you understand that, the process becomes clearer, not as a trend, not as a claim, but as a system following the same rules that govern everything else in the physical world. Energy, structure, and the constant effort required to keep order intact. That is the foundation, and everything else follows from it.